Up next, we have Laurinda Nyarko, a Ph.D. student in environmental engineering. Laurinda's talk is on studying the impacts of heavy metals in water, a new method. Laurinda, welcome to the stage.
Thank you, Karina, and thank you all so much for being here today. I'm really excited to share with you what I've been studying. And for the past 4 years, one of my projects has been developing a new method to study how metals interact with organic compounds in water. So why are we interested in this? Here's an EPA report listing some of the things that contaminate our surface waters. As you can see, metals are third. These are metals like copper, lead, zinc, and others. And now some of you are thinking, that must be concerning. And it is. And several studies have shown that even small amounts of these metals can harm aquatic life. This brings me to my next thing. And that is when metals get into water, they like to interact with things like organic compounds. Basically, metals chemically stick to organic compounds to form metal-organic complexes. These complexes often account for more than 90% of the chemical forms of metals in water. But the challenge is, we don't know what these organic compounds are because we don't have the right method to do so. But finding a method is really complicated because these organic compounds come from a lot of sources. Some come from nature, like when plants decay, and some come from things we use every day, like cosmetics and detergents. These can all end up in our waters and can all potentially bind with metals. That is why it's important to know what these compounds are. The method we've developed is a combination of liquid chromatography and mass spectrometry. Here's our LC, which we use to separate the complexes. And we do that by taking a small amount of our sample and injecting that onto this column, which is packed with a specific material, and a complex is separated by interacting with the material on the column. But at the end of this process, we have a bunch of separated complexes, but we don't know what they are. That's what we use the mass spec for. And this is what makes our method novel, is that we use two mass spectrometers. This one quantifies the metals, which tells us how much is there. And the other one identifies the organic compounds that the metals are binding with. The first step in a mass spec is ionization, which simply means we apply some energy to charge the complexes. Then they go to a detector, where they produce a signal that we can record. This signal is based on the mass to charge ratio, which is a unique number for every individual molecule, and that's how we can determine what the complexes are. So that's it. That's our new method. And for the next few minutes, I want to show you how we've used this method to identify copper complexes in wastewater. And our main goal here was to determine if the wastewater treatment process changes the complexes there. So wastewater is actually one of the ways that heavy metals end up in our surface waters, and it also has a ton of organic compounds. So it's important to understand what happens once they get to the treatment plant. We collected 3 samples at 3 stages. First, after primary sedimentation, this is pretty much untreated wastewater. Next, after secondary sedimentation, after biological treatment, where microbes break down many contaminants. And third, after disinfection. This is the water that goes back to our rivers. So let's look at what happens after the first mass spec. On these plots, on the y-axis is the intensity of the copper complexes, which is how much is there. And on the x-axis is the retention time, and that's how we're able to pair the data from both mass specs. This overall curve is the shape of copper that is binding more with natural organic compounds. But the more a peak diverges, the more likely it's binding with man-made organic compounds. Every individual peak is a different copper complex. And the higher the peak, the higher the concentration. So what this tells us is that in the untreated wastewater, copper binds with both the natural and man-made organic compounds. So then what happens after biological treatment? As you can see in the orange curve, the complexes look different. There are fewer peaks, which means the treatment process has removed some of the complexes, which is a good thing. But there's some new ones. And the overall shape of the curve is shifted. What this tells us is that as the microbes are breaking down the contaminants, they're also creating some new organic compounds that are also sticking to copper. Now, this is where things get exciting, because we can use our second mass spec to start to determine these changes. So for example, that tall peak in the orange, what is that? We can overlay the data from both mass specs based on that retention time I told you about, and then using that M over Z value, we can predict a molecular formula. And we can even determine a structure of the molecule. An interesting fact, this chemical is actually used in the dye for blue jeans. So it could totally end up in wastewater. So what happens after final treatment? As you can see in that green curve, that tall peak is gone. And there are basically no individual peaks on the curve, which tells us that at the end of the treatment process, what remains in the wastewater are these complexes that are binding more with the natural organic compounds. So let's go back to our complex. We're no longer confused because we have more information on what they're made up of, thanks to this pretty awesome method. And thanks to it, we've been able to identify about 3,900 molecular formulas. And we're trying to determine structures for these molecules. But we can actually use this method for other metals and even other water sources, like the river, which would be good to know so we know what happens once the wastewater is discharged back into it. Ultimately, we hope this tool can be a valuable resource for other researchers and government agencies like the EPA to regulate metals in our water, to protect those water sources, protect our aquatic friends, and protect us. Thank you.